now is our last presentation of the day, Stephan Kemp, who will be talking about genetic insights and biochemical lessons in ALD newborn screening.
Thank you, Elisa, for having me again this year. It's really nice to be here again after, I think, 3 years. The last time was before COVID. I'm going to tell you something about the Dutch newborn screening program and the lessons we learned by evaluating, the numbers that we got out of the screening and also from our diagnostic testing. These are my disclosures. Everything I'm going to tell you is being funded by the Ministry of Health and supported by ALD Connect and the Virgin Tillman Foundation. Like your state, New Jersey, we also nominated ALD in 2013 in the Netherlands. My colleague Mark Engelen and I, we did that because we heard that there were plans to expand the Dutch newborn screening program. But it took 10 years before we started screening live. And part of it was because there was a challenge. The Dutch Health Council recommended that only boys had to be screened for ALD. And there's no program in the world for any disease that has a test in which you can only screen for either boys or girls. So we had to develop that. I will not go present all the details from the study, because I've done it at previous meetings. So I'm just going to summarize it in 3 slides. So the challenge we had was to design a screening algorithm that identified males by only using dried blood spots. Because this was mandatory or recommended by the Dutch Health Council, so it was not my decision. So we developed a 4-tier program, in which was based on the algorithm from New York State. But as a second tier, we included what we call the X counter, in which, based on DNA, we just counted X chromosomes as a way to identify males. And our 4-tier program includes ABCD1 sequencing. So if there's a referral, it comes with a C26 lyso PC and an ABCD1 variant. And another part of the challenge is that we have to finish this within 35 days, because 35 days after birth, families will always receive a notification, whether the screen is positive or negative for any disease. Of course, it's a positive screen, they will be notified earlier. So in 2021, we performed a pilot in 4 provinces in the Netherlands, here shown in the 2 colors. We screened over 71,000 newborns, and we identified 4 boys with ALD. And also, before we started, we also developed a prospective monitoring and long-term follow-up protocol, which I show on this slide. It's fully operational. And one of the important things is that we decided, because with ALD there's no immediate hurry to notify the family, is they want to make this as positive as possible in this situation for families. So what we developed is that once the family is notified, and in the Netherlands that happens by the family physician, already an appointment at the Amsterdam Leukodystrophy Center is scheduled. So the family is notified, and within 24 hours they will meet with my colleague Mark Engelen. And then, of course, the blood sample is taken to confirm the diagnosis. And then the follow-up program starts. And the families come to the clinic on Wednesday. They see the pediatric endocrinologist, they see the clinical geneticist, and they see the pediatric neurologist. And this continues every 6 months. And then at the age of about 2 years, the MRIs will be taken. And because the screening was so positive, we had to write the report and everything. Nationwide, the Ministry of Health decided to add ALD to the Dutch newborn screening program. So starting October 1st last year, all boys in the Netherlands are screened for ALD. But what else did we learn? Because we had access, we were a pretty good organized country. And if you have your paperwork and your questions right, we have a lot of committees. And so and from coming to these meetings, and we discuss at the round table, and we ask questions. So we know that there are some questions that are out there, and we have the option to investigate these and to answer these questions. So when we finished the pilot, we received anonymized data from all the newborns that were screened. We received several covariates, and we could use these for analysis. And one of the important things is the cutoff for tier 1 and the age of sampling. So one of the questions that's out there is, what's the effect of gestational age on C26 lysa PC? And since we had the information of gestational age and the C26 lysa PC at tier 1, we could plot that. And if you can see, the C26 lysa PC is very constant throughout in embryo development. And if we plot then the ALD cases, we see that they're nicely elevated. So the important lesson here is that there's no need to rescreen a preterm newborn for ALD at term age. I think that's a very important message. So we investigate many more covariates, and so we can conclude that C26 lysa PC is not affected by either gestational age, sex, or birth weight. So the little yellow dots in the middle are the ALD boys, and they're sitting nice in the center of the cloud. Another important thing is confirmatory biochemical testing. And now we're talking about the HPLC method or the plasma variant chain fatty acids. So this is not the tier 1, this is the dedicated method. It's important to realize that plasma variant chain fatty acid levels are false negative in 50% of women with ALD. And that may have consequences for newborn screening. So it is possible that the C26 lysa PC results maybe have normal plasma variant chain fatty acids in case it's a girl. And it's shown here on the slide. So you can see the women in pink. Some of them have normal variant chain fatty acids. Our laboratory in Amsterdam decided around 2018, 2019 to see if we could switch to C26 lysa PC. So to align newborn screening and diagnosis of patients. So always use the same biomarker. Another reason is that it's very tedious to do plasma variant chain fatty acids. And C26 lysa PC is much easier to do, much quicker, and it's also, as far as we know, it's not affected by the diet. And importantly, in some cases, in many women with normal plasma variant chain fatty acids and ALD, they have C26 lysa PC that are elevated. So it's a superior diagnostic test. And we published a paper in 2020, and if you're interested, I'm happy to share that with you. This is just a little show-off. This is our Ali Fatima. He visited us 3 weeks ago, and he wanted to make a selfie. So this is not the selfie, but so we have a combined. The laboratory in Amsterdam is a combined diagnostic lab with over 30 years of experience in diagnosing peroxisomal diseases, mitochondrial diseases, lysosomal storage diseases. But we also a research lab, and these are our mass specs. The ones in the back are all for diagnostics, and the big one here on the left is a purely research machine. It's a timsTOF. And the nice thing is, because we are a combined lab, we are ISO certified, so there's not everything we can do. I'm not allowed to use these machines. So we have operators for this, technicians for that, and the researchers are not allowed to use these diagnostic machines. But as I said, over 5 or 6 years ago, the lab changed to measuring C26 lysa PC, and since we do a lot of diagnostic testing, and we have access to the data, we can do some other investigations. And one of the important findings is that the C26 lysa PC level is higher in newborns compared to non-newborns. As you can show here, these are controls. And you can see that the mean of the newborns is higher than the non-newborns. So this is important for confirmatory testing or for children that are screened at older age. And C20 lyso-PC is a part of the erythrocyte membrane, so we think that this is likely due to the erythrocyte concentration, which is high in newborns, and it drops after birth, and it more or less levels out after 3 months. And so if you look at this graph on the right, and here we see one ALD patient, confirmed ALD patient, and we measured it at newborn age. Then at the second dot is the confirmatory testing after 1 month. And then the third dot is a sample that was taken after 3 months. So this was earlier than the 6 months. But the family came to the clinic after 3 months for the first time already. And we were allowed to take blood samples, and when you measure the C20 lyso-PC, you see that they drop. So it's very important to know the age at blood sampling for confirmatory testing. And we have different cutoffs for newborns and non-newborns. And I would like to switch to genetic testing and then specific variants of uncertain significance. So it's very important to realize that there's a clear distinction between symptomatic diagnosis and population screening. In diagnostics, often a patient visits the doctor. So this patient, this individual, has clinical signs and symptoms. And if these point towards a disease, often a blood test and a laboratory test, or biomarker test, or genetic test can confirm the diagnosis. This is rather straightforward. But if you move to population screening, the entry point is not a clinical question. The entry point is a biomarker, as shown here on the left. And how do you establish a cutoff for any test? So, as the previous speaker mentioned, often you take 1,000 or 5,000 or 10,000 random samples, control samples, and you measure the level of the biomarker, and then either at the upper level of controls or some other statistical trick you can use to set up your cutoff level. But what do we know about the cutoff level? Is the cutoff level that's not equal to the disease level? So there is this gray area in between, referred to the gray zone. But if you set your cutoff level and you start population screening, you screen a lot of samples. You will identify samples that are above the cutoff level. And these are then screen positive. And because they're screen positive, they're sequenced. And it's very likely you will find an amino acid change that is causative of this biomarker to be a little bit elevated. But the question is this high enough to make you sick? So these are these orange dots there in the gray zone. There will be a variant in the protein. So there is a small change in the protein, and it affects the enzymatic activity, and the biomarker will be a little higher. As I said, the question is it disease causing? Is there any benefit for the newborn, or should we identify these variants? And when you do a little bit more analysis, I'm keeping track of mutations and everything. And another interesting finding is that since 2015, there is in the United States, there is an annual rise in the number of newborns that are screened for ALD. But there's also a rise in the number of VUSs identified. These are the gray bars. And when you put these graphs together, you see there's a nice correlation. So what happens often is that a family following newborn screening receives a genetic report from often a commercial company. And very often the conclusion is a variant of uncertain significance, and then the variant was identified in the ABCD1 gene. Yeah, now what? What's next? A VUS is a variant of uncertain significance, but it often creates a very unclear situation. And last year, together with Troy Lund and Joe Orsini, we wrote an editorial on this. Unfortunately, it then ended up behind a paywall, because I was in a fight with a Dutch publisher. But if you want to have a copy of this, I'm happy to share it with you. It's a nice, easy reading. And One of the tables, this is a table from the paper, and one of the conclusions that we could draw is that the probability of finding a VUS in newborn screening in ALD is about 3 times higher than prior to ALD newborn screening. So, and as I said, the finding of a variant of uncertain significance creates a diagnostic dilemma. Often these are missense variants, so there's one building block, one amino acid is changed. It's by definition not previously reported, otherwise there should be more information. And yeah, so there are no clinical clues because we are doing population screening. So we screening people or individuals in the pre-symptomatic stage. And yeah, and there is no family history of disease. So the question is this VUS disease causing or are we creating patients? And this is not new. This is not unique for ALD. In almost any population screening program, there's a rise in the number of VUSs identified. So here for some of the metabolic diseases, Pompe, Fabry, MPS 1, but also for cardiovascular screening, for breast cancer screening, you see that there's a rise in the number of VUSs that are identified. And this is, again, this is not unique for ALD. So this is for Gaucher disease. In the pie plot, the gray area are the VUSs. So 49% of the variants reported in ClinVar, so this is data from ClinVar, are VUSs. This is for mitochondrial dystrophy, 66%. For CFTR, it's even higher, and also for breast cancer. And if you're still not convinced, these are several other diseases that I just investigated. So basically for any disease, for any gene, the majority of the results currently in ClinVar are VUSs. And there's a steady rise in VUSs in ClinVar. Again, this is plotted over the years. And what is clear is that the VUSs that are reported in ClinVar rapidly outpace the pathogenic and benign assignments. So this data is until 2022, and so I looked at the data for October last year, and by now it's over 85% of the missense variants in ClinVar are VUSs. So we have a problem. So the problem statement in ALD is that here, in ClinVar there are, well, this was a few months ago, 1,129 missense variants, of which 500 are missense variants. And if you think about it, you can say, okay, this is 500, but if you think about the story behind it, it means that over 500 individuals and families and their treating physicians received a report from a genetic often company saying that, yeah, we have no idea. I think we have to do something with that because the consequences of this are pretty high. So it often results in parental stress, anxiety, what to do. There will be additional tests will be done or maybe treatments. So there's a financial burden for the families. There is an increased cost in healthcare, medical harm. Is it necessary to do an MRI? Is it necessary to do another test? And if this continues in this pace, I think the scary thing is that eventually legislators will say, well, what's the balance between health and cost? So, and it also may result in reduced trust in screening programs. And of course there may be a risk in decreased participation and reduced effectiveness of the treatment of the screening. And if you look at the literature, there's an increase in the number of publications that deal with VUSs. So these are just some of the publications that I picked up. But I think in ALD we can do something about this, and this is one of the tasks that I set out to do. Because the first question you have with a VUS is this pathogenic or is it benign? I think that's a very important question. And luckily in ALD we have a database that I started in 1999 with Professor Hugo Moser when I was still at Kennedy Krieger. And here I'm holding a baseball bat of Cal Ripken. I also play baseball, So I knew the baseball bat was over there, so I wanted to hold it. And the ABCD1 variant database is actively maintained and curated. So I am in contact with a lot of physicians, and if something new, important development is, it will be updated within 24 hours or instantly, depending on what I'm doing at that moment. I think the important other part of the database, actually I have to say thanks to Elisa. The first time I was at a meeting, she asked me, this database, is this from the university? Is this accessible? And then I realized that, okay, I'm here now in the newborn screening community, which is very different than the patient's visit to the doctor. And just a simple table with all the publications. So I decided to completely rebuild the database. It took me about 2 months as a side project. I dig up all the literature since 1993, and I scanned all the papers and started counting cases. So, clinically affected patients. I'm not reporting phenotypes here, because the genotypes have no predictive value for clinical outcome. And we know that, but there are many people looking at the database, and they may not know that. Of note, there are 200,000 visitors per year for the database. I have no idea where they all come from. Yeah, they come from all over the world, but why there's so many? So the remark section is I think very informative, because it tells you something about the variant, how many cases there were with all the references, but also if you look at scientific papers, a lot of papers use cell lines. If you use cell line and you mention the mutation in that paper, and you show that there is no protein on the Western blot, or there's no beta oxidation in the fibroblast, you can connect all of the information. And that's exactly what I did. So for some of the variants, there's more information known, so the one at the bottom. But most diseases, so I also noticed that many ALD families think that every disease has its own database. No. I think there's only one, CFTR has a database, and ALD, and I think there are no other ones out there, at least no other ones that are actively maintained. So most diseases, they rely on ClinVar, and the classifications that are given in ClinVar. And this is just another comparison. So in the ABCD1 variant database, there are over 2,500 missense variants. And in ClinVar there are 1,129, and you can see in ClinVar there are many gray ones. But if I compare the data from ClinVar and ALD, I can curate the ClinVar database. That means that a lot of entries in ClinVar are not correct. And I think it's a black box. We have no idea who at that company decides if it's pathogenic or not pathogenic. There are, for the sake of time, I threw out a few slides, but you can just see that there's sometimes the classification is wrong. So I think that's also another problem. So as I said, the key question is a variant of uncertain significance? Is it pathogenic or benign? And I also noticed that sometimes the uncertain is misinterpreted. I'm in contact with people, and then they say, well, it's uncertain whether it's causing cerebral ALD or myelopathy or adrenal insufficiency. But no, that's not the U. The U is it pathogenic or is it benign? So we need to start at the beginning. And there are ways to address the pathogenicity of a variant of uncertain significance. So we can do biochemical confirmatory testing for pathogenicity. And I think most important is collaboration. And that's another unique thing in ALD. Most ALD researchers, clinicians collaborate. Many of us know each other for many years, and we like to drink a beer together, and that always helps. And we like to make fun of each other, that always helps. And so there's information sharing, and by that you can connect the dots. And this is an example. So here we have newborn screening, and the clinician, and then the database in the center. So this is an example in which a variant was identified in newborn screening, and it was never seen before, so we classified it as a variant of uncertain significance. It is identified in newborn screening, but it has not been associated with disease. Then it was identified another 5 times, but it remained a fuzz. Then I was contacted by one of my colleagues that a patient was at the clinic with clinical signs of ALD. So here we have a clinical endpoint. The C26 lyse-PC was elevated and the same variant was identified. Okay, then you can upgrade the variant to likely pathogenic, because now there's a clinical case. And of course, it's also always possible that an error is made, so in one case, we say it's likely pathogenic. So now in the future, if another colleague calls me, or emails me, and says, well, I have a patient here, and it's the same variant, then we can upgrade this variant to pathogenic. So, and I think upgrading to pathogenic is not that difficult, because we also have the biochemistry. The other way around is much more difficult. And for that we started the Gray Zone project. And the Gray Zone project is funded by ALD Connect and by the Toulmin Foundation. And it's based on what happens in real life. So here we have an example, newborn screening, first is identified. So here we have the newborn. Then clinical genetics starts drawing the family tree. And many times there's an uncle or a grandfather with the same variant. Often these males, there's no clinical problems, and then suddenly, okay, now I have ALD. So I think we can use this, because if these males have the same variant, and they are seen by one of the ALD specialists or neurologists, and the clinical assessment rules out ALD, or maybe very mild spinal cord involvement, and we collect a sample, we can use these samples to do further testing. So we've set up a system, it took us about 3 years to set up this logistical system, and really with the help of Kelly Mitunen from ALD Connect, we have it now in place, and I will show you some results. If a blood sample can be taken by the local clinician, and we have now, ALD Connect and the Toulmin Foundation from the Gray Zone project, we will pay for shipment of the blood sample, well, not plasma, because it's very expensive, but the dry blood spot, by FedEx. So if you contact ALD Connect, you get a FedEx sticker, and also newborn screening cards, and an envelope. So the blood cards can go in there, and then they are shipped they go by airplane to Amsterdam. And in Amsterdam we use our cool machines to analyze C26 lyse-PC. And as I said, the advantage is that we've been doing this for 5 years, so we have an enormous large data set. We know the disease range, we know the control range, so we can use the blood spots and we can measure the C26 lyse-PC to get some insight into the pathogenicity of this VUS. So officially the Gray Zone project started 2 years ago, but in hindsight we have already analyzed samples from all the countries here in green. And this is some data I show. I've not plotted the variants here. Because we're still in contacting some of the families, and I don't want to disclose that before they know it. But here in green at the bottom are all the controls. These are over 1,200 controls, and this is control data from 2021, so I'm going to update that. The green circles are variants that for which we know they are benign, because they're very, very frequent in the general population. And we had access to some of the samples. So yeah, you see a green one there that is a little high. But the majority of the benign ones are at the bottom. And you also see that some of the gray ones are clearly benign. I lost the pointer here. There's some here in the normal range. But you can also see that the majority of the VUSs, they are here at the bottom. And all the red dots are plasma samples from ALD patients, so we know these are levels that are pathogenic. But you can still see that between the green area and the pathogenic variants, there is still this gray zone. So Unfortunately, still a lot of the buses are in the gray zone. But you can also see that many of them are clearly in the pathogenic region. So these buses should be pathogenic. Yeah, so we will continue to do this, and I'm telling all my, so I'm not just interested in buses, I'm also interested in plasma samples from proven patients, because we also are analyzing C26 lyso-PC and connecting it to clinical outcome. Because I haven't shown it today, but we also see a clear correlation between, well, pretty clear correlation between the level of C26 lyso-PC and disease severity. And we have a paper now that's under revision, and we are just expanding. So we received a lot of samples from Minnesota, from Boston, from Leipzig, just to keep validating and adding more data points to this. So yeah, so in the end, what do we expect to find and how will we act on this? So yeah, it's pretty clear. Vessels will be reclassified as likely benign or likely pathogenic. And also, as I showed in population screening, the clinical spectrum of ALD will expand. We eventually will identify through extended family screening males that at the age of 60 or 70 have mild spinal cord involvement. So if we always link these to the same pathogenic variants, the question is, should we pick up these variants in newborn screening? Is there any benefit for the newborn? So in my ideal world, there will be a list of variants for which we know they are mild and late onset, and then yeah, and then of course it's always up to the states what they want to do with it. But I know that very likely in the Netherlands, these will be flagged as do not report. But of course you need to have a lot of data to support these decisions. I think I mentioned the third point now. Yeah, so of course the ultimate goal is to have cutoff levels that match risk stratification. And with that I mean, ideally in newborn screening, you want to pick up the boys that are at high risk of developing adrenal insufficiency and through ALD. And I think the biochemistry is now coming more and more closer to showing that we should be able to do that. And for that, years ago, Joe Orsini already started a harmonization project, and to be honest, in the beginning I had no idea what he was talking about, because I said every variant in ABCD1 is pathogenic. Well, how wrong was I? And that's one of the things we talk about at the roundtable meeting, because almost all states have different cutoff levels, different machines. Some use a homebrew test, others use Perkinelme or Revity. And yeah, I think ideally you should, and of course tier 1 is a very difficult analytical method. C26 lyso-PC is sitting in the Revity kit, but actually it's a molecule that doesn't like to be in there. But okay, we just have to deal with it. So eventually the bad thing is that you would like to harmonize all this. And so one of the decisions that we made in the Netherlands, going from the pilot to the live nationwide screening, is that in the pilot we noticed that everything below 200 nanomole in our tier 3, your tier 2, were VUSs, or no, there were no variants in ABCD1. So we decided when we go nationwide screening that we already raised the cutoff for tier 3. And I preach this so often to the public of health in the Netherlands that I was the project leader of the pilot, so my role is not there anymore. But I convinced them that in the Netherlands we can do these kind of studies, like the ones I showed, the gestational age. So they asked me to monitor for at least 2 years in the nationwide screening to do the validation the measurements of the screening, and to look at all the data with the aim to eventually increase the cutoff of tier 1 and possibly tier 3. Yeah, the acknowledgement. This is there's not a single person. I think all of us in the room are to be thanked for this. This is truly a community-broad project in which we all work together, and I think that's another nice thing about ALD is that many people think that all scientists and clinicians are like that, but it's I sometimes are at another conference, any other disease, a mitochondrial disease or so, and then or they come to our disease and they say, You all talk together. You're not competitors of each other, and I think that's another very powerful thing that we can be proud of. And these meetings are a perfect example of that. Thank you.
I had a question, and I'm not sure if you mentioned, how many samples have you collected for the gray zone?
Oh, officially, since the logistics started, 12. But we already had a lot of them in the freezer, and they were technically also gray zone samples. But it's now the logistics are finally sorted out. It's increasing.
Anyway, so just a comment. Thank you. That's very good. The first thing about the burden, when you had to list the burden, another burden, what we find is conditions that are serious. If you tell a parent that you have maybe a serious condition, that also changes their family trajectory. They're not going to have other children if they think they have a child that has a serious illness, just because they don't know if they take care of them. So that's another thing you didn't involve there. I think that also plays in. Yep. The other question I have is, in terms of the variance of unknown significance, there's beginning to be data out there for the individual conditions, because as you know, a variant it's not equal across genes. We see variants that more likely will become benign, and others that will have a higher incidence of going the other way. So, for example, they're starting to do this with craniofacial conditions where they see actually the numbers are very low. They have others where the numbers actually are quite high. So the missense mutation in one gene is going to have a different risk in another. So I presume over time we're going to have to wait a long time, but you'll start to get data on how many genes go one way versus in this gene, how many missense mutations go one way in this gene versus the other. So that at some point you'll be able to say 20% in our database of missense will actually become pathogenic. That's one thing. The other thing is, I don't understand why you wouldn't, for example, wouldn't it be easier, because we do this commonly, again, I'm not an expert in ALD, but if we have a mutation, we would first do, and the companies really do this very cheaply, do the parents. So if it's a new mutation, then it's probably real. If it's in one of the family members, you have it. Why would you want to send it across the world? The individual companies will do that for you for $50.
Well, benign variants also are inherited. So if there are no clinical signs.
Yeah, no, but it's more likely. In other words, you're pushing
true.
it. A new variant, yes, it's not a SU, but it's more likely. And that's what we do in clinical counseling.
Well, the simple answer is it's not mandatory to send it to Amsterdam.
I'm sorry?
It's not mandatory to send it to me. It's sure, there are other ways, but in case I've been in contact with families that have been pursuing their VUS for over 5 years. Yeah. So I agree with you, but often it appears not to be that easy. Yeah, because in my world where I don't have such a thing, if I have a variant of uncertainty, I would first do the parents. Yeah. Again, the new patients, if it's it's more likely to be real, more likely. The other thing, though, is I can do all the family for $50 in a private company, and then have that data, and then follow the expression of the gene myself. In other words, I don't need. Why wouldn't you just put a consortium together to pay? That's what happens here. This is what happens often. So now you have your family tree, but yeah, but why wouldn't? But I'm saying sending the blood over to a one center. You said that the family member's blood could be sent somewhere. That's often what happens. Often what happens is that they draw blood and then they send it to a CRO or a company to have plasma variant chain fatty acids. And then it comes back elevated. The problem with commercial companies is that they can set their cutoff levels for the controls. But commercial companies don't have patience. So, and then often I hear, I am elevated. And I always make the joke, you're elevated. We're talking about DNA now, aren't we not? Yeah, but like I said, how elevated do you need to be to become sick? And go ahead.
The problem is that I think your logic works best in a condition is de novo. In this condition, a significant proportion are inherited, and that's true for
Right.
the pathogenic and the non-pathogenic.
Agree. But what I'm saying is, in terms of actually following it over time to actually determine whether it's real or not, and there you're looking at clinical evolution, right? Expression. So it's going to be hard in this because the expression is so age dependent. Now, what I'm saying is that my understanding is you have a consortium where family members' DNA are sent. Is that true or is that not? Is that true?
The C20 is Lysa PC, yeah. And we compare the results with over 1,500 controls and over 300 ALD patients.
But your study, doesn't it collect DNA from family members?
The variant is already known because that's how the whole thing starts. There's a fuss.
So you are?
Yeah.
I'm sorry, I don't hear very well. Are you collecting DNA from family members? Is that part of your...
No, that's always often. So here, this is often what happens: is that family receives a notification of a fuss. Then the clinical geneticist or whoever will make a family tree and identify other males with the same variant. And then you can send these in for plasma variant chain fatty acids, but if you send these into a company or that doesn't have disease levels, it comes back elevated. Well, what does it mean, elevated?
No, I think maybe we'll talk later.
Yeah, I think that's a good idea.
Excuse me. I would like to know a little bit more about the instrumentation you use for the mass spec. Which make it is? Any idea of that?
Oh, we have Waters, Bruker.
Okay.
Yep.
Okay, and I assume your matrix is dried blood spot, or is it the plasma what you take?
For the gray zone, we do blood spots, dry blood spots. In the publication that I showed, we compared dry blood spots and plasma.
Okay.
And for reasons I don't understand, there's a straight correlation between plasma C26 Lysa PC and dry blood spot.
Okay.
Which is, I don't understand it, but it's very handy. So we can use dry blood spots and plasma for this.
And what will be your quality control material in that case?
Sorry?
The QC materials or the quality control materials?
In this test?
Yeah.
Oh, our laboratory, our diagnostic certified quality control samples that we have.
Okay. And one small thing, like if you we get the mutation testing done from any lab, and we got a report like probably it's a VUS or a benign. So is it good idea that we again verify the same mutation in the database what you use? And sometimes the result will be biased, and when we don't know like which way to go like the
Trust my database.
Okay.
Yeah, like I said, for ClinVar, we have no idea who is calling the classification.
Yeah.
In the past, it used to be diagnostic labs that are associated with Hopkins or with our institute, or in which a physician would ask their diagnostic lab to do DNA testing. So the data was connected. Now you send it out to a commercial company, but the commercial company doesn't have the clinical data. So it's disconnected. And then somehow somebody says, okay, puts it in ClinVar and gives it a classification, but we do not know on the basis of what.
So do you think that in future the companies might be coming up to your database for their result prediction?
I know some of them don't, but I don't. I've always questioned why. Because it's free?
It's an open source, I think.
Yeah. Yeah, again, if you go to adrenoleukodystrophy.info, it's there.
Thank you so much. For
Oh, you're very
all the
welcome.
Would you mind going to the slide with all the dots, with the green dots on the bottom, and then the...
No? This one?
Yes. So I'm having trouble understanding it. So some of the gray dots are even a little above some of the red dots.
Yeah.
But they're still in the
they are
gray
gray
zone.
dots because these are versus. These were variants of uncertain significance. So it was not known if they are pathogenic. So we tested them. And the ones that are in the red area, they're clearly pathogenic because the C26 Lysa PC is in the disease area. So even though in that case it may be de novo or it may be, I mean, you know, the penetrance ALD is strange. I mean, you can have a child with cerebral ALD and you can have an uncle with late onset. So there may be 50 years apart before clinical symptoms start. So if you measure a plasma sample or a dry blood spot and it's over there, it's pathogenic. But we call it likely pathogenic because to be really calling something pathogenic, you need a combination of clinical proof, biochemistry, and genetics. And still, what you're missing here is the clinical proof. But it doesn't matter for follow-up if it's likely pathogenic or pathogenic. It's the same follow-up procedure.
Okay, thank you.
One more question over here. Thanks.
Oh, there.
Oh, yeah, sure. So, what about other genetic modifiers beyond ABCD1 variants? And because I can imagine there'd probably be other genes that could have variants that could have an impact on VLCFA levels too, especially in the context of a mutation or some variant in ABCD1.
That's a perfect question. Yeah, yeah, absolutely. So the C26 is the product of the peroxisomal beta oxidation, but it's also it's made in cells through elongation from long chain fatty acids. But you can also have desaturation. You get a oxidation. So it's a combination of all these enzymes, and all these enzymes may have variants that doesn't make you sick, but do affect the enzyme activity, and that contributes to the final concentration. So there's no genotype-phenotype correlation, but it looks like that the biochemistry is much more correlated to the disease severity. But of course, if we talk about modifiers, it's not all biochemistry. There are also so we only have identified one modifier yet, but it's expected to have modified genes in maybe inflammatory genes. And yeah, I mean, the field has been looking for these modifiers in 1993, and we only identified one gene, which is not predictive, and that's an enzyme that's involved in omega oxidation. And interestingly, also in leukotrienes, and leukotrienes are inflammatory, so that was a very interesting one. But yeah, we are about, I think later this year we will do whole genome sequencing on all 160 Dutch patients. And Mark Engelen, my colleague, he's been following these patients for 8 years, so we have really deep phenotypes. And we have patients that are over 50 years age of old, 50 years old. They only have spinal cord involvement. They don't have adrenal insufficiency. They, based on MRI, have no brain involvement. And so this is the mild, extreme. And on the other side, you have the boys with cerebral ALD and adrenal insufficiency, and that's the most severe form. And so if you have enough of these patients, and you can use those patients for whole genome sequencing. And another very interesting group are, I didn't show it, but there are boys that develop cerebral ALD, but their C26 levels are lower than the other cerebral ALD boys with adrenal insufficiency. And my simple thought is that these boys developed cerebral ALD because they have a very unfavorable or unfortunate composition of the modifier genes. So these boys will be maybe the clue to finding these modifiers. Because if you have enough of those, I suspect you will find variants in those genes that are causative for a higher risk of developing cerebral ALD. So again, this is collaboration. I think a lot of patients, it's a rare disease, so not a single center has enough patients. So if you all collaborate together and you really clinically assess the patients and make sure you have a good biobank and yeah, and then collaborate, I think that's the way to move forward. And I think what the last 3 years what we're doing, actually since we moved to C20 Lighter PhD, is that things are starting to make sense. I've been working on ALD for 30 years, and I sometimes joke that we're really good at making hypotheses, and then we go drink a beer, and then we go to the lab, and we do one experiment, and the hypothesis is out the window. And for the last few years, we're starting to predict things. If you do research and you predict things, and the experiments come back and they fail your hypothesis, that means that you're coming closer to the truth. And that's what's happening in the last 5-6 years. And
So the C26 LPC is it looks like it's going to be a nice biomarker and has been so far. Can you just comment on how consistent its levels are, like, let's say, within the same patient at different time points?
they are.
Within the same family, so like men in the family, let's say, new baby and grandfather, and then across families. So with the same genotype, in other words, because we have, now a few situations where we've got lots of different family members, and so how tight are these things gathering?
We have some VUSs, for which we now have about 9 samples, and the 9 measurements are really close together. They're really group.
And is that unique among the other fatty acid metabolites? I mean, is LPC kind of, is it the most consistent of those, and that's why you've continued to use it, or do the other fatty acids, just in this context, also show consistency across?
You mean lipids, or which other fatty acids?
Yes. Yeah, the other very long-chain fatty acids like C26.
Yeah, but then the complex lipids like PC. Yeah, we have a whole bunch of biomarkers that are really, really powerful. But you have to keep in mind that lipidomics is not a targeted assay. The power of this assay, it's a targeted assay. So we have an internal standard for C20 lyGPC. And for every metabolite, if you really want to do diagnostics, you need an internal standard for that specific particular molecule. So, yeah, we are thinking of doing an ALD fingerprint. We have several PC species and lyGPC species for which we probably need to make internal standards and set up dedicated methods, so that you have a set of 8 or 10 lipid biomarkers that you can use to get more confidence. And hopefully they're even better than the single one. But in the end, it will not just be lyGPC. As mentioned, there will be genetic markers also, because it's not clear cut yet. It's getting very close, but this is not just the answer. It's helpful, but we need to go on. I'm thirsty.